This is the third lesson of Chapter 11. We're going to be further talking about Mendelian genetics and starting with Punnett squares. Punnett squares are a tool that can be used to predict the outcome of a genetic cross between two individuals that you would know their genetic makeup for a particular trait. So let's start by looking at an example related to Mendel's experiments with the pea plants, having one parent that was purple flower and the other parent with the white flower. Now, we would need to know the genetic makeup of those purple and white flower plants, and we'll talk a little bit more in the next slide about why that is, because there could actually be two different options for a purple flowered plant for their genetic makeup. The one shown here has two of the dominant alleles, or they are said to be true breeding, homozygous, or pure breeding, while the recessive individual, the white one, has only two little r's, which are the recessive alleles for white color. So although it shows the outcome of the first cross, all of the offspring are heterozygous, having the dominant allele from the purple flowered parent and the recessive allele from the white flower parent. This does not show the Punnett square though. So for the second experiment that Mendel did, we have the Punnett square shown here. Remember that he allowed his F1 generation offspring that only had purple flowers to self-pollinate. So the genetic makeup of the parents of that cross would both be big R, little r. And that's shown in the Punnett square below. Across the top would be the genetic makeup or the combination of alleles for the plant that was providing the pollen, serving as the male. So capital R is the column header for one of the cells in the Punnett square, and lowercase r would be the other column header. For the other parent, which would be the female donating the ovules, she would have the same genetic makeup. Capital R for the dominant allele for purple, and lowercase r for the recessive allele for white. So in the resulting four squares would be the combinations, all possible combinations of those alleles. So big R in one, the first top left box, big R in the top right, big R in the bottom left, little r in the bottom right. Now, this could be interpreted as, again, a ratio for making a prediction about the outcome of this genetic cross. This doesn't mean that there's only 4 individuals from this cross. In fact, it's saying that 1 out of 4, or 25%, would be big R, or the term is homozygous for purple color. 50%, or 2 out of 4, would be heterozygous, big R, little r. And 1 out of 4, or 25%, would be little r, homozygous for white color. So this is how we can use a Punnett square to make predictions, and those predictions are quantitative in nature, as percents or fractions. Now, I did use some terms that we might not have seen yet, but they will come up in several other examples when we're talking about Punnett squares and the genetic makeup of organisms. So the word homozygous here is defined as individuals having 2 identical alleles for a gene. Another term for homozygous is true breeding, or pure breeding would be another term that you might encounter as well. Heterozygous would be the other scenario where you have an individual with 2 different alleles for a gene. So in the case of the one shown here at the right, let's apply what we've just talked about to this. So the purple flowered plants that have both the dominant alleles, big R, would be homozygous. But also the white flowered plant would be homozygous because it has 2 recessive alleles. The heterozygous individuals would be the ones that have the dominant and the recessive allele, big R, little r. And Another term you might see for heterozygous is hybrid or not true breeding. Hybrid would be really the term you would see if it's not heterozygous. Okay, so in addition, when we're interpreting the outcomes of these genetic crosses, we sometimes want to know about the physical appearance, like whether the flowers were purple or white, and those are the phenotypes. The physical appearance, the term for that is phenotype. And so as we're studying different other examples of Punnett squares in genetics, we will focus on the phenotype, but also the genotype, the genetic makeup. In these cases, we're talking about the combinations of the alleles as a simplification. So the genotypes here in this scenario would be big R, big R, big R, or little r, little r. Now there's going to be times when you can see the physical appearance, the phenotype, but you might not know the genotype of that individual. And in fact, this affected Mendel in his ability to know whether or not the plants that he purchased at the market to work with were true breeding. Because recall that the purple flower could be from, actually you see it here, the genotype for a purple flower plant could be either homozygous, dominant, big R, or heterozygous, big R, little r. So how would one know if the plant, the purple flower plant, is indeed homozygous dominant or heterozygous? Well, there's a way that can be determined through something called a test cross. Here's an example of a test cross that might have been a useful tool for Gregor Mendel when he purchased his purple and white flowered pea plants from the market, not knowing whether the purple flower plants were homozygous dominant or heterozygous. So as shown here, it would be the dominant phenotype with an unknown genotype, big P, or big P, little p. Versus the white flower plant would only be the genotype little p. There's no question about that. So what he would have done and what you could do in a similar scenario is you take that dominant phenotype and you cross it with an individual with the recessive phenotype. And there's two different scenarios that would come about here depending on what the dominant genotype is. If the dominant genotype is big P, as shown on the left, then the outcome of that cross would be 100% purple flowered plants. However, if the dominant phenotype was heterozygous for the genotype, big P, little p, then as you see here, there would be a 50% or 1/2 of the offspring would be purple and a 50% chance or half the offspring would be white. So really, if even one individual offspring had white flowers, then it would be known that the dominant individual carried the recessive allele for white flower. So this tool, this test cross method, can be used in other Punnett square scenarios besides in Mendel's, and we'll see that later as we do different types of inheritance problems. Another type of cross I want to talk about briefly is a monohybrid cross, which in fact we have actually seen an example of this, but the term hadn't come up yet. Monohybrid refers to when you have one trait and you have two individuals for that one trait who are heterozygous or hybrid for that trait, and they're being crossed. So this would apply to Mendel's second experiment. He did a monohybrid cross when he let the F1 generation offspring, the ones that had the purple flower, but they actually had that hidden recessive allele, and he let them cross. And then he saw in the F2 generation offspring the reappearance of that recessive trait. So here's another example related to Mendel's pea plant studies, where we have in this Punnett square scenario, capital Y, which would be the round seeds or the smooth seeds. That's one of the parents in the original parental generation, crossed with the recessive individual that has dented or wrinkled seeds, little y, that's recessive. All of the offspring of that cross would get one of those alleles from each parent. So they would have capital Y, lowercase y. they'd be heterozygous or hybrid. And so when allowed to self-fertilize, they have the same genotype and they're both heterozygous for that one trait. So that's why this is called a monohybrid cross. Notice that the outcome of this is a 3-to-1 ratio, where for every 3 that have the dominant phenotype, round or smooth seeds, there's 1 that has the wrinkled or dented seeds. So that's actually a tip, a hint. You can always expect the phenotypic ratio for a monohybrid cross to be that 3-to-1 ratio. And I made a note about that here at the bottom of the slide, that the phenotypic ratio for a monocross is always going to be 3-to-1. Now that we have established what a monohybrid cross is, in our next lesson we're going to look at an example of a dihybrid cross and experiments that led to Mendel's law of independent assortment.